Hi everyone, welcome to my channel or welcome back to my channel. So this video is the second video on gene editing. The previous one gave some overview of how gene editing works, in particular CRISPR-Cas9, which is the current state of the art of gene editing. In this video, I want to address what it can be used for currently, as the current technology exists to address different genetic diseases. Okay, the major limitation of CRISPR-Cas9 is that in nature it's used by bacteria to create genetic mutations, and this is to protect bacteria against viral genes. But in human medicine, we want to fix genetic diseases rather than create mutations. So most of the time, CRISPR-Cas9 is actually doing the opposite of what we want. However, there's a couple instances where it can be useful as it exists right now. Okay, a couple of these are for addressing dominant diseases and also permanent exon skipping. And then I'm going to talk about an adaptation of CRISPR-Cas9, which can be used to create some true single base editing. Okay, so dominant mutations, so what this is are a mutation where if you have a single copy of a mutation, you have a disease, which is opposed to recessive, where you need a mutation in both copies of the gene. So in a dominant disease or a mutation that's associated with dominant inheritance, and for many genes there are mutations that cause dominant inheritance or mutations that cause recessive inheritance. Okay, but a dominant gene mutation, if you inherit from just one parent, that's sufficient to cause the disease. Okay, so why is that? Well, there's a couple different reasons. Maybe you simply need two good copies of the gene. One isn't enough. It doesn't make an adequate amount of the protein from just one copy of the gene. But another one is, what if it produces a protein, but the protein behaves in some way that it's not supposed to, and wouldn't if it had the correct sequence. Okay, well, so gene transfer therapy, which is basically just putting a new good copy of a gene into your cells, that can be used to address the first disease mechanism, the same as it would for a recessive disease where you have no good copies of the gene and giving one. If you need two good copies and you already have one, then you need one more, and then everything's good. But it's not going to help with the second one. If you're making a protein that causes a problem, putting another good copy of the gene isn't going to fix that, because you've already got a bad copy that's making a problematic protein. Okay, so this illustrates how this works. Okay, so you have a normal gene, and it makes a protein, which is a string of amino acids, and it folds together in a certain way. Then say you have a mutation in your other copy of the gene, and it causes a variation in the protein sequence, and say the protein isn't folded the right way. It does something it's not supposed to be doing. So that you'd actually be better off if you just didn't make that uh, abnormal protein from the gene with the mutation. Okay, so the way you could, in theory, correct this with, gene editing, CRISPR-Cas9, is actually you already have a mutation in the gene, but it still allows you to make a protein. What if you just break it completely so you can't make any protein at all from that copy of the gene? Then you're just making the normal protein. And let's say that this protein, you can make enough from just a single copy of the gene, to be healthy, have no problems. So actually, uh, causing a break in DNA is actually a positive step. It inactivates a harmful protein, which is very much how bacteria use CRISPR, when in that case the harmful gene is for a virus. Okay, so then the second category I'm going to talk about is what's called permanent exon skipping. Now, to explain what that is, I first need to explain what exon skipping is. Okay, so a gene basically contains portions of DNA called exons, which actually code for the amino acid sequence of the protein. But they're not in a single gene all continuous. They are interspersed with non-coding regions called introns. So the first thing you do when you're expressing a gene to make a protein is you copy the DNA into RNA. Then that's your working copy of the gene. Then you splice together the exons and remove all of the introns. Now you have just the coding sequence for the protein, and then once you've spliced the RNA, then you translate that into amino acids. Okay, so DNA and RNA have a certain sequence of nucleotides, marking the beginning and the end of each exon so that splicing can occur. Okay, now say you have a mutation and it's going to be in the coding region um, in a certain exon. And say that this mutation is such that it either completely prevents a protein from being made, or it's so badly garbled that it's unstable. Okay, so what if you could just skip over this exon? uh, you're going to be, missing, a portion of the spliced RNA. You'll have a smaller protein that's going to be missing a certain amount of amino acids. However, for some genes and some mutations, exon skipping, is actually a therapy that's used today. Okay, so the way that current exon skipping drugs, and there's a couple, I think now 3 now, exon skipping drugs that are approved for Duchenne muscular dystrophy. It turns out that the gene for DMD, for various reasons, is particularly amenable to exon skipping. Now, the current drugs work at the RNA level, so they don't change the DNA. They only change the working copy, which every time you're expressing the gene and making a new batch of protein, you make the RNA. But the RNA doesn't last for very long. The DNA is actually the archival copy of the gene. So you have to keep taking these exon skipping drugs. Now, what if you could, by CRISPR-Cas9, get make cause exon skipping to be done permanently? Okay, and the way you do that is you call well, I skipped over the slide, but basically you either erase or, for the current RNA drugs, you mask the nucleotide sequences marking the beginning and ending of certain exons. So then when the cell starts doing the splicing, it doesn't recognize that, this portion of DNA is actually an exon. So it just skips over it. Okay, so the third thing I'm going to talk about is what's called base editing. Okay, so there's four DNA bases. They're abbreviated A, G, C, and T. And in this case, I've written out the full names of them. A, T, C, and G are what they're commonly abbreviated by. Okay, and you'll notice that the chemical structures of A and G are kind of similar to each other. They both have two rings. And the chemical structures of C and T are similar to each other. They both have one ring. Okay, so A is always paired with a T and G is always paired with a C. Okay, so some very clever people figured out a way of partially inactivating the Cas9 protein, and playing some other tricks so that you can switch between an A and a G or between a C and a T. However, you can't make any of the other possible substitutions. You can't change an A for a T or a C, for instance. Okay, so we'll go over a common mutation in the FKRP gene, which is responsible for LGMD2I, or in the new nomenclature, R9. And this is actually by far the most common single mutation in limb-girdle muscular dystrophy. So what this is a C to an A at a certain location in the DNA sequence, which causes amino acid number 276 to change from leucine to isoleucine. Okay, so okay. However, base editing only lets you change an A to a G. Okay, so I actually had a conversation with a friend of mine who's working on, base editing, and we was like, well, what mutations might we be able to? And we both thought of FKRP, and sort of got really excited until we realized that, well, you need to, change either the A back to a C, or it turns out changing it to a T, which isn't the sequence that normally exists, but that would also change the coding back to leucine. So unfortunately, that isn't correctable. There are a number of other mutations that can be corrected by this approach. Although they aren't nearly as common as this one I'm talking about. And it turns out also that the efficiency of base editing as it currently exists kind of depends on the DNA sequence nearby. So it's not even that you can change any C you want to a T. There have to be kind of other, sequences of DNA nearby for it to work well. Okay, so there are some things that we can do now. There are a lot of people working on DNA editing, but it's not quite ready for prime time. It's still in very early stage, so some patience is going to be required, for it to have a major impact on treatment of muscle diseases.